Hello. Yeah. But at the same time, I need to give credit to all the speakers who are following behind, all my fellow colleagues and friends for pitching in this synthetic biology thing, which is really a paradigm shift in the biotechnology. So I'm going to talk about synthetic microbiome, redefining host-pathogen interaction. And I come from Loma Linda University. This is a medical university with 18 different colleges. We have to credit many patents and many other important things. We were the first people to do the heart transplant. What you see down here is Dr. Leonard Bailey, who is actually the person actually in charge. So, coming to the synthetic biology, biotechnologists are always indeed very capable of solving problems. But at the same time, we are also getting very accustomed to getting better in solving problems. So these two are very important for a scientist to come. Starting from sequencing genomes, earlier sequencing genomes were a very big pride. And then after that, the scientists found that there were a slew of data where they were very confused with a very big voluminous bulk of data. So reading them into a useful, readable frames were very hard. Then came the next problem, editing genomes, and then editing according to the useful readingness of the protein coding sequence, non-coding sequence, etc. As I've told you, solving problems and getting better in solving problems. Now the world is totally focusing on writing genomes. This is where the synthetic biology mainly lies. So I see that this FTP program is a combination of Department of Chemistry and Department of Immunology. So if you ask a chemist, he would tell that the whole life hinges around the four important elements or four or five important elements. But whereas if you just talk to a biotechnologist or immunologist, he might focus on the live DNA. But at the same time, I should certainly say that it's only a small stretch of DNA that is going to make a difference between a sunflower and a small baby, or between a small grown grass to a very big grown tree. With these kind of variations, people started focusing on a stretch of DNA, and they wanted to see what that stretch of DNA were doing. And one of the important fascinating discoveries was the green fluorescent protein that you would see here, where out of all the whole genomes, they took a chunk of sequences that code for this green fluorescence. And then now you all know that this is being very much used further. So what is then synthetic biology? And what is the difference between genetic engineering and synthetic biology? Synthetic biology is a very big umbrella where you can create new entities, new bacteria, new life, or specific biological parts or devices, or any new systems, or even you can define the whole networking, the metabolome of the system. So everything comes under synthetic biology. So genetic engineering is a very recent topic, and now we are still focusing on synthetic biology, where the computer science people become married to a biotechnologist. That is what I should certainly say. So now the big question is, can we make life fully engineerable? So this is $1 million question that many people would ask now. And can we make life programmable so that we can program our life accordingly, so that a 70-year-old person can look like a 20-year-old person, or a person who is totally tired of running because of gasping be able to run as an athlete? Can we reprogram them? Can we engineer these things? Or can we synthesize even new life? These are all very big questions in biotechnology nowadays. So as you all know, that this century is called as the century of biotechnology. We initially started with exploring biotechnology. A lot of inventions were there, right from invention of discovery of DNA to restriction enzymes and etc. And now, as a biologist, we have to be turned into a designer of biology. We need to design biology. And then not alone to design biology, we need to build in biology. This is what the synthetic biology takes place. I should talk about the iGEM, where the International Genetically Engineered Missions competition that happened in Stanford, where they gave to 12th grade or 12th standard people a fascinating topic to synthesize life. And this iGEM is a very big program that was very fascinating, where the people had such a lot of brain minds they bring in to these kind of new life programmable things. So when you see on the other side, now you would see here, this is the 2010 Science cover page. This was a news by Dr. Craig Venter, who's a very renowned molecular biologist. So he has first created the new synthetic life, which is nothing but the synthetic life with 482 coding proteins and 32 RNA genes. With these Haemophilus influenza, where they found that totally this particular thing can be programmable and this can be synthesized without any lively matter into it. Later on, Craig Venter went on to use this kind of a process in using them to the brewer's yeast, which we call it as Saccharomyces cerevisiae, to produce human proteins, to produce human skeletal proteins, etc. So this was the forerunning concept of a synthesis of a new life, where this was given a very big impetus. But whereas this is talking about a single bacteria or a single entity, but whereas if you talk about a human microbiome, this is an admixture of many, many microbes that are there in our body. Many live in our skin, many live in your mouth, many live in your nose, many live in your stomach, vagina, etc., everywhere. So if you just take 90% of them are bacteria, and these 90% of bacteria can be synthesized, this is a very big problem here. So then what happens is people delved into this human microbiome and human microbiome project throughout the whole world. The reason being people wanted anything new, so they moved on from an older concept to a newer concept. So every country moved on to these human genome projects, where in 2007 human microbiome project was launched to study what are all the microbes that are present in your mouth, in your gut, etc. And in 2008, after seeing USA, the European community also joined along with China. The reason being to identify the species richness of the bacteria in your body, because there were a lot of corroborations to say that this species richness was the main culprit in making a person fatter, or for the diabetes or any inflammatory diseases. The reason being the biome or the bacteria, probably that I would say here, is flooded with lot of energy, and each microbe species in the biome, they can transform this energy into a new molecule, where we are not bothered about what kind of a molecule that produces, whether that molecule that produces by the biome inside your stomach or inside your mouth or inside your nose is going to cause you a problem or an advantage. So this was published in 2015, which gave a very plethora of all the viruses, bacteria, fungi to the phyla level. But whereas this did not say anything about whatsoever what does a microbiome cause. Then later on, people went on to study individual microbiomes, and they found that the most diversified microbiome were found in the mouth as well as in the gut. The reason being that mouth and gut are interconnected, the bacteria can travel. So henceforth, now from here, I will call bacteria, because the whole the topic hinges on the bacteria. So this is a map of diversity of human microbiome. So in the human microbiome, you have two kind of bacteria or microbes. One are good in nature, another one is bad in nature. So as you know, in life, everything hinges around good and bad. So for example, lactobacillus species, which are good, they are present in vagina, mouth, gut, etc. But whereas there are other detrimental bacteria like corynebacteria, bacteroids, campylobacter, they are also very detrimental in here. So this was published in Trends in Genetics, which gave a very great impetus of focusing on certain bacteria to study what they really do into the system. You might ask me, why do we talk about this in a synthetic biology talk? But I'm here to tell you that we are focusing on a couple of bacteria because the whole world, as you're seeing this slide here, there are a lot of universities that have jumped into this human microbiome thing and even into synthetic biology. So, what is the microbiome effect that people are very worried or people are very fascinated is that they totally change the whole human physiology. So, the bacteria that lives in your mouth or in your gut is going to transform you to be very healthy or not healthy, aged or very young looking, etc. So, microbes are now called as an organ totally, like your hand or your leg. The body also has another appendage which is called as a microbiome. So the metabolic capacity is very huge because there are over 3 million genes from these bacteria and viruses, and they are thought to have a great function more than the liver that it does for the human being. And also gastrointestinal bacteria, GI bacteria, where they are very much important because they are involved with the metabolism part. Because a person cannot live with the food, and whatever the food that we take is going to vary from one place to another, or from one species to another, or even from one race to another. So these produce metabolites at the end, and these metabolites are very important, and whether that is beneficial or that could be reprogrammed is what the synthetic biology of the microbiomes focus on. So what can a microbiome do? The whole microbiome, I should say that we have 3.3 million distinct genes, unique genes, 150 times more than that of the humans. For example, say 60,000 genes to 3.3 million genes in the microbes. 10% of the human transcriptome is regulated by human microbiota. This is a very big focus here because the epigenetics plays a very vital role in modifying the human being based on this 10% of the human microbiota, and they affect primarily the epigenetic means. So, for example, a person who can smoke is predisposing to many diseases. Like that, the microbiome that lives in your body also predisposes to many, and also they affect the energy metabolism, carbohydrate, etc. On the contrary, they also are very much helpful in strengthening the immune processes, reducing the inflammation, and producing many good substances like short chain fatty acids, mucins, BDNF, which helps in neuron production, etc. And with all these good in nature, people wanted to focus on good bacteria. And the reason being that if there are no good bacteria, the host passes from a symbiosis to a dysbiosis, where dysbiosis is an imbalance of the bacteria between good and bad. So, what makes a very big important characteristics here is the diet. What we take inside predisposes to what kind of a bacteria that lives in our body or outside the body, and that is more important. And if there is going to be dysbiosis, and then they are linked to many diseases like diabetes, even GI disturbances, even mood disorders. There is a study which says that people who take McDonald's cheeseburger, they get very irritated. So these are all certain research findings which are very fascinating. So when you just see the normal healthy intestine versus a dysbiotic intestine, you would see here there are only less number of bacteria where these bacteria are the main culprits of causing the disease, and they are called as keystone pathogens. So not all the pathogens are called keystone pathogens because these keystone pathogens are the king of pathogens. So they decide what the other pathogens or other microbes have to do, and they are involved in many diseases. So what we did in our lab is that we took a double pronged synthetic approach. It's like taming the bad bacteria, making the bad bacteria become good. And on the contrary, we were also doing empowering the good bacteria. We selected a couple of bacteria and we made a synthetic approach of empowering the good bacteria, the best bacteria in the system. So we focused on keystone pathogens of the microbiome. And we mainly focus on the metabolic function and the immune function of these keystone pathogens. I'm going to talk about is called Parvomonas gingivalis, an anaerobic bacteria that lives in our mouth as well as in the gut. Versus empowering the good, we were targeting on the beneficial microbiome in modulating the metabolic circuitry of the synthetic probiotic bacteria, making them synthetic probiotics, or making a couple of bacteria synthetic prebiotics where they can predispose to the metabolite that will bring about more probiotic bacteria to propagate, such as lactobacillus species here. So what you see here is the keystone pathogen that I was talking about, Parvomonas gingivalis. So one bad thing or good thing about this whole bacteria is that this is called as a king of bacteria in the microbiome, and they are the keystone pathogens, one of the keystone pathogens. When you just see the whole operon architecture, you would see that the operon which I'm just talking about are basically collectively called as the virulent modulating operon. So this is the main key of this keystone pathogen to make this bacteria either good or bad or worse or whatever it may be. And this out of which the main key, the knob of the key is the VIM-A or virulent modulating gene A, which is mainly involved with a lot of other functions that you see here. So I don't want to go very deep into these functions. What I'm coming to say here is this operon is more important. So what we thought was to do a synthetic approach to modify this operon so that it can be modulate the keystone characteristics of this bacteria and make them calm and quiet. So other than that, we also wanted to focus on the virulent modulating gene E, F, and AROG. They are all aromatic. They focus on many other things. For example, AROG is a chorusmate mutase, which codes for chorusmate mutase, and this is it is used in the metabolome of producing an aromatic amino acid. So with this operon structure, the genome structure, with this particular operon, we found this operon to be very fascinating, and we focused on in modifying them on a synthetic approach. Why did we choose VIM-A? Is just because this is on your left side. What you see here is a very a comparative analysis of many bacteria, many keystone bacteria, where you would see here the VIM-A or the VIM-A ortholog plays a very vital role. What we did was two different type of approaches. Number one, we did a chimera of VIM-A. It means to say that we took the VIM-A gene and we put it into another location on the same genome. So that chimera had two copies of VIM-A. So to say that this bacteria, the chimeric VIM-A P. gingivalis, will have more virulent modulation. At the same time, we also did a double mutation to see what really happens if we are going to take that away. So that VIM-A, if it is going to be taken away, does the other operon takes the place of that VIM-A? We did a metabolic manipulation of this thing to see what happens in these two different stages. What you see on the right side is a genome map showing the major modulatory genes. What happens when we take the gene out or when there is going to be a chimera. What you see here in dark blue or black are the common normal unperturbed genes versus the ones that you see in blue and violet are the ones that are highly modulated. We wanted to just take this genomic data, and then we wanted to see what is a metabolome or what is a pathway that is mainly affected. What you see here on the left side is the anabolic pathway that is being carried out inside a bacteria versus a catabolic pathway. So we also went into the flux analysis of this synthetic metabolic pathways. So we took the strain, the chimeric strain, and we wanted to see what is the total variation in the expression of certain metabolites in bringing about certain metabolic pathways or the circuitries which are more important for virulence. So what you see here in the red ones are the highly upregulated expressional circuitries. As you can just correlate this with each and every metabolic pathway which I'm just talking, where you would see here majority of the whole system of the bacteria. What you see here is the whole system of bacteria, and you would see here all the things, both catabolic as well as anabolic, they are being affected here. On the contrary, what we saw was there was a total null stop with the bacteria that is being devoid of that particular gene. And there is no other gene in the operon that is taking care of that particular women. You could see here it is minus 2.1, where you would see everything in yellow, versus the ones that you see in red, which are more than 1.1 plus. So this manipulated metabolic pathway by the chimeric and the mutant strains were easily identified, and we thought that we could take this as a very important step in doing a synthetic circuitry in modulating this bacteria. So we totally sketched what are the modulatory pathways or the metabolome that is being affected. What you see in red are the highly upregulated pathways. And we are going to focus on the tryptophan, alanine, and other things. And also, we are going to focus on the substrate pyruvate or in glutamate or acetate. Because when certain bacteria become very mischievous, they switch on the substrate from pyruvate to butyrate, and then from butyrate to pyruvate if they become good. Likewise, the amino acids are also very much modulated, because these amino acids, ultimately the whole system produces certain important molecules in the system, metabolites. So this is a comparison between the chimera as well as the mutant. But what we found very interesting was that both the strains had one big modulation that is being very much affected, which is the amino acid metabolism. And we just wanted to focus on this amino acid metabolism, especially the bacteria, and what are all the major functions that a bacteria does in the case of modulatory function. What we found that siloglycoprotease, or a post-translational modification called sialization or a sialidation, that is being affected. This is more important because the bacteria needs this post-translational modification when they want to become very mischievous, and they can affect the immune cells. Likewise, the production of the small vesicles to a very thick cover, glycocalyx, or to identify the lipopolysaccharides, alteration of lipopolysaccharides, to making a very thick cover that protects the bacteria from other invasion until expression of the fimbriae. So when we found that many of the important modulatory functions are being affected by this synthetic method of transformation of this detrimental bacteria, because I should certainly say here, when you talk about synthetic biology, many people, they do not focus on a very bad bacteria to make them good, rather than they will focus on good bacteria to make still more good best bacteria. At the same time, taking WIMA, we also wanted to just focus on another important PTM, sialization. This is more important because this post-translational modification is very much needed for the bacteria to interact, either gram-positive or negative, to interact with any cell in the human system. The reason being because of the terminal sugars that are there in the eukaryote, the human cells, as well as in the bacterial cells. So we took three important genes to make them synthetic and manipulate them. These three are mainly focused on siloglycoprotein or siloglycoproteases, which can modify this particular end sugars, which are nothing but the N-acetyl muramic acid, or we call it as sialidases. So we took these three strains. We made three strains, out of which we made a chimera as well as a mutant. And we did the whole thing like we did it for the women. The reason why we did this was because that is the time, and that is the area, the superficial area of the bacteria where they interact with the human cell like macrophages and other things. So immediately when a bacteria goes inside your body, the first line of defense produces an important lectin called a Siglec, which is nothing but the sialic acid binding lectins. Many Siglecs are there, out of which Siglec 11 and Siglec 16 are very important for the inflammatory response. So what we thought was, when we are going to modify this one by means of synthetic means, by the circuitry of producing the siloglycoproteases, we can modify and make that particular bacteria very calm and quiet. So to say, to make a lion to become a cat. That is what we were just thinking. And we also did the same thing, and we actually did a great deal of identifying them with to study the interaction between different siglets. I don't want to go very deep into these things, but what I am coming to say here is we did find that when we modify that siloglycoprotease by synthetic means, we identified that there were entirely different interactions between the macrophage and the bacteria. It means to say that sometimes the macrophages immediately gulp the bacteria and the bacteria were dead. So the dead bacteria is gone. Sometimes the bacteria they live in the system, but they are totally not at all causing any problem and they live in the system. So this is what you see here, the surface sialidase interaction that you see between the host pathogens. And we did an orbitrap mass spectrometry. We just chopped all these terminal ends of the carbohydrate. We wanted to study them. And we wanted to study them, the reason being, what is unique of these sugars that help them in the host pathogen interaction. So we did a high performance anion exchange chromatography, which is a very high class glycan identifying system. And what we found was there were a unique pattern of sugars that were present in the human neutrophils, also in P. gingivalis cells, in all the cells, in the outer membrane vesicles or in the cells or in the supernatant secretions, etc. So to say, there were very unique pattern of interaction which we identified. So this unique pattern can be cut off by modifying the synthetic circuitry of this sialidase or siloglycan protections. So initially what I was just telling was we identified the virulent modulating genes. The second one we focused on genes that are immune modulating. So now we made two strains through synthetic means by little modification where we identified the uniqueness of these strains. When they are put into systems like mouse, they did not produce any disease, but at the same time they were helpful in nature in nurturing the good bacteria. Coming to the good bacteria, the beneficial gut microbial community can be classified into two different types. For example, firmicutes and bacteroids. For example, if a person is going to be very obese, then we could say that firmicutes predominate your stomach. And if a person is going to be lean, we might have seen our own friends. People will eat lot, but they will be always thin. But whereas a person who eats 2 idlis more a day will become so obese. The reason being the gut microbial community that makes this fascinating variations. So this is one of our study where we identified what are all the microbiome that exists in the different cohort study in Africa, Oceania, South America, Asia, North America, between health and disease state with teenagers, elderly, infant, adult, etc. This is a very huge study with a lot of people into consideration. What we found was, we found the bacteroids, the P. gingivalis, the keystone pathogen, that was found in all the 6 continents that were analyzed, that you see in blue. So now you know why did we choose this keystone pathogen, P. gingivalis. And this P. gingivalis was now being documented, now evident, I am showing you here as a keystone pathogen. Let alone this, we also wanted to see the metabolic circuitry. What happens if the bacteria is present? How the bacteria causes the other bacteria to behave wrongly. So we focused on another group of bacteria called Odoribacter, which also modified the substrate in which the bacteria could dwell. So what you see here is Odoribacteria, and what you see here is the ethnic group like African American, Asian, Caucasian, Hispanic, etc. And what we did here, we mainly focused on this Odoribacteria because of the substrate they produced. So with this background in nature, we wanted to just classify what are all the bacteria that are present in a good human being, healthy human being versus a diseased human being. We found a set of bacteria which are blue in color that are found in healthy, but a set of bacteria that are present only in the people who have diseases, are prone for diseases. And also there are certain black colored ones which are existing in both the kind of things. So here is the thing. We wanted to study the differential metabolic expression of these genomes. So as I have told you already, we focused on the bacteroids as well as we focused on the Odoribacteriae. And we wanted to see whether we can modify the circuitry to switch the substrate between butyrate to pyruvate. So this metabolic expression, what I'm just showing in yellow, are the major metabolic variations that were found between the healthy as well as the affected individual, diseased individual in this microbiome that we have studied very extensively. So we focused on the whole metabolic pathway of butyrate as well as the pyruvate to see what we can juggle with in modifying them. So what you see here is an augmented positive host pathogen interaction, where you would see here there are a lot of other bacteria that have the same metabolic pathway. As I've told you already, that the bacteria and the human being, everyone has the same metabolic pathway, but the bacteria, they have little miniatured one. And you know that these all the bacteria have the same kind of a lysine sequencing, lysine metabolic pathway, but ultimately they end up in butyrate. So what we did was we focused on this metabolic circuitry to modify them, and we took certain bacteria like Parphomonas gingivalis, and we modified the genes here to make a circuitry so that the butyrate is not being produced. Why the butyrate should not be produced? The reason being is so simple that when the butyrate is there in the system, sometimes what happens is this butyrate becomes the feeding background for many bacteria that causes diseases. So then how do we change them? See, how do we change the bacteria that live in your stomach? It's because based on the dietary diversity or what food we take. You know, there are 75% of plant genetic resources that have been totally lost now. When you take, we only eat 12 plant species and 5 animal species for our food. Other than that, we don't eat any other thing. We don't eat snakes like Chinese people. We don't eat lion meat. So we eat only 5 animal species, or we eat only 12 plant animal species. That is more important here. So then what kind of a diversity that we are talking about here? So then plant versus animal diet, they came a very big issue here. As I did not tell you that Loma Linda, the place where I come from, is a place which is under the blue zone. Blue zone is a place where people live more than 100 years. There are 5 places in the world, out of which one is Loma Linda. So what we did, this plant diet versus animal diet, was not done in any mouse or in any animal lab animal model. We did it in human being, and we found that plant-based proteins play a very vital role in increasing certain important beneficial bacteria versus reducing certain detrimental bacteria. So they increase the short chain fatty acids against, if you are taking animal proteins, trimethylamine oxide, which is being very highly upregulated or being produced in your system, ultimately causes the cardiovascular disease. So we also did study between the Western diet, gluten-free diet, Mediterranean diet. We found Mediterranean diet to be very good, and you could see certain species of genus of bacteria in here. Okay, we went into to study what is the abundant beneficial species that exists in our system. So what we did was, you could see here, Lactobacillus cryptopsis, Propionobacterium, Streptococcus, etc. Every bacteria that I'm just showing you in this round, they're all very good bacteria that are being present in all the microbiome. See, you can take anterior nares, that is nose, to buccal mucosa, to saliva, to throat, until stools, feces, to mid-vagina, etc. So now you see here, now we have a group of beneficial bacteria, we covered them, and we found them to be unique and existing in all the population. Okay, so then what is the point in we taking this beneficial bacteria? So what is new in them? What do they do inside the system is more important. You know, this bacteria which sits in your whole microbiome, no matter if it is going to be hair microbiome, nostrils, skin, stomach, etc., they produce important metabolites, such as small molecules. And these important metabolites are very enormous, like 1 into 10 to the power of 30 in the whole biosphere that is being produced inside your microbiome. But whereas when you just take small molecules of important microbiomes per se, there are certain important therapeutics that are being produced by these kind of, for example, lacticillin by Lactobacillus graciae, epidermidin by Streptococcus epidermidis. So these are all being categorized, but at the same time, these are more than this. There are a lot of uncategorized small molecules. So what we did was we took the microbiome. So we took 100 species, we mixed them. versus monoculture, one, like a keystone pathogen or whatever. And we did a slew of mass spec proteomic analysis to find out what is the unique spectra that identified unique small molecules that we have not found so far. So this, what you're seeing here is, for example, these are all certain bacteria that produce certain small molecules. For example, FN stands for Fusobacterium nucleatum. PG stands for Paphromona gingivalis. See here, PG stands for Paphromona gingivalis, etc. So we found certain unique small molecules that we identified. We did a comparison by bioinformatic analysis to find out what kind of a categorized small molecule they belong to. And finally, this is a total outcome of the secreted small molecules that we found in the microbiome by each and every bacteria that we found very important. For example, the Paphromona gingivalis keystone pathogen, they produce a small molecule called methyladenosine-like molecules, which are very detrimental in producing cancer. But whereas if we are going to just modify that circuit, we can take that particular circuit to a very beneficial way. Likewise, as we are talking about beneficial bacteria, we focus on lactobacillus. I don't want to mention the species of lactobacillus here because we have submitted a patent on this group of bacteria. So they produced a putative 3-indole propionic acid, IPA-like molecule, which is very good for the health. How it is very good for health is just because that this IPA is an ultimate sequence of the metabolite in tryptophan metabolism. So please recollect my earlier talk, part of the conversation where I was just focusing on tryptophan amino acid that is being not modified by the keystone pathogen. Now you see here the ultimate endpoint is IPA, and this is very much effective. For example, this I am just showing you, this IPA is very much effective against Mycobacterium tuberculosis. So now here is the thing where we take the microbiota, and then we focus on the tryptophan circuitry here. We modify the circuitry by synthetic means so that then we modify the bacteria, lactobacillus species itself, and then we can use it as a synthetic probiotic, and it could be used for the human being. What you see here is a little more elaborate sequence of the circuitry that has been synthetically modified, where you would see here the shikimate pathway or the aromatic amino acid pathway that finally ended up in tryptophan. So what you see here is upregulated gene versus downregulated gene. You modify these things to find increased tryptophan production, and from tryptophan you would be able to identify, you will be able to get the IPA, which is a very beneficial metabolite that is that you could identify, that you could get it from lactobacillus. So here it is very simple that you take the lactobacilli species of importance, we modify the circuitry of shikimate pathway, and we identify the overproduction of tryptophan when it is needed in your system. When it is needed in the system, when we are affected by certain important diseases, like say Mycobacterium tuberculosis or Clostridium difficile, etc. So we came running down into a couple of species, which I'm just giving you here, where you would see that this is a very extensive study that we have done. And one of our collaborators had a very good article in Science, where he had taken up the microbiome from the Hasta hunter-gatherer tribe people, where they are having the best diversified microbiome in the whole world. And now study is underway with these Hasta hunter tribes, and also on the counterpart on the blue zone areas in Loma Linda, to identify what are the unique bacteria that make you live longer, where you can modify them synthetically. So how do we program? For example, we go for the microbiome, we take their microbiome, we identify the bacteria, okay, this is good bacteria. Then how do we program them to produce only good things? So we program them, the basic skeleton I'm just giving you in the synthetic biology of synthetic microbiome production is identify a sensor and a signal here. For example, a signal is any detrimental bacteria that produces a metabolite. That metabolite has to trigger certain sensors. Sensors are nothing but certain stretches of DNA which are called genes. They will produce protein, and these proteins will attach to another indicator, And then when these two get attached here, they will trigger the RNA polymerase to produce a very good set of genes to be producing good set of proteins. Okay, having known this signal, sensor, etc. So how are we going to just program the bacteria? That is going to be a very great important thing, which we call it as a gating mechanism. How do you do is that it is based on the repressor proteins that we have already studied or compared to a promoter protein. You need to know that when a repressor protein is there focusing on one area and sequencing on one direction, it is going to be a repressor. Versus if it is going to be the same repressor can be a promoter in a different system. For example, the input that makes this sensor trigger should bring about this particular gene to produce an output. And if the input is not there, the output should not be there. So, okay, this is a single circuit that we are going to go. But whereas in a bacteria, you have more than some 5,600 circuits or more than many circuits that should be there. We have talked about, two component system, three component system, different mechanisms of modulations, etc. So then we can put on input 1, input 2. For example, you can put a repressor, you can put a promoter, where the promoter becomes a repressor here, and a repressor becomes a promoter if it is going to be on the first. So then, now the question is that how are we going to identify such a lot of repressors and promoters? So this is where the bioinformatics came into existence, where there are a lot of repressors that are being totally aligned to a promoters to identify whether they are unique or not. So that this one, input 1 and input 2 should be entirely different and they should not be identical to make the output on or output off. This is what we call it as a gating mechanism. And this gating mechanism is helpful in bacterial program. Why? Because we see these kind of repressor sequences where we identify them and what is this repressor going to focus on, which gene. We have this idea, and with this particular biological data, we go and make them to an electrical circuit. So what you see on the right side is an electrical circuit here. It's like a switch here. Imagine when there is going to be a chemical here, there is going to be switching on here, switching off here. When there is going to be a chemical B here, there is going to be a switching on here. So when you have this circuitry, this is like an electrical circuit board that you would see in any computer chip or whatever. So rather than making it in silicon, you do it with DNA. That is the main thing, and that is the main crux of the synthetic biology. So these are all switches, biological switches that are made by pieces of DNA. How do we know these pieces of DNA sequences? Yes, we have already studied them in the repressors. So getting a database of the repressor promoters are more important to design these kind of switches. So one kind of an input will produce one kind of an output positive. Another kind of an input becomes negative and it will off the whole circuit down below. So then what we do, after we do a logic minimization of this thing by different gating mechanisms, we add different repressors, etc. We add it from the database. We generate and we do this circuit. Okay, we just give it them for a printing of DNA and other things, where we identify the sequence of gating mechanisms, and then they are actually produced. These DNA sequences which produce these gating mechanisms like 1, 2, 3, 4, they are aligned in such a way that these DNA sequences are given for construction here. So now what happens is, you have to write a program in the computer. So here the computer science person comes into play. There are a lot of programming methods by which we can identify the database sequence. We have a separate database, and then we can create a switch here by means of computer algorithm, make genetic circuits. Once the genetic circuits or the gating mechanism is being produced, the whole sequence is being given, where the circuit is converted into the DNA sequence and they are being produced. And this DNA sequence, when they are being produced, they can be taken inside a bacteria, where this bacteria can be programmed to a synthetic bacteria. So what I'm telling you here is a sequence in which certain mechanisms that exist biologically can be converted into an electrical sequence of switching on and off mechanism by which they can be put inside a living system where they will act accordingly, because the switch is not electrical now, but it is only the DNA sequence here. So a couple of examples which I have given you here. For example, a traditional microbiome. For example, if you are going to eat a probiotic, say lactobacillus tablets, that will have only single input. Rather than if you are going to eat a synthetic beneficial microbiome, they will have multifaceted variations that could produce each and every different unique input here. For example, if you are going to take a synthetic microbiome, for example, diet. Imagine people who are used to taking more of carbohydrate diet. We need to know that even eating more carbohydrates, they in certain kind of feeding habit, if you have a rhamnose, this is more important. It is highly inflammatory in nature, where there should be an integrated memory array where there is the same switching mechanism should be there, where this should be totally stopped. It means to say that rhamnose should not be metabolized, but they have to be taken out from in the species, in the feces. Likewise, metabolites, which I was just talking about, many metabolites that are there. For example, the metabolite that we are talking about is a common inflammatory metabolite like nitric oxide. Imagine when the nitric oxide level is higher in your system inside, it means that there is inflammation in there. So these metabolites like nitric oxide, when they are present in excess, they will trigger certain switches by which the recombinase, for example, this is a recombinase switch, this recombinase switch will switch on that particular bacteria to produce a very new metabolite or a very advantageous metabolite or small molecule that will help in reducing the inflammation. That is what I was just giving you here. So this is the main focus of this synthetic beneficial microbiome now. And people have come out with a lot of beneficial microbiomes. For example, like P. acne, the bacteria, are being wired in such a way that they produce an antibiotic when you are going to just put the same bacteria as a cream here, where you will not see pimples. Like the lactobacillus species that is present in the vagina. See, people who have vaginal discharges and other things, when they are being given a suppository of this kind of lactobacillus bacilli by synthetic means, they could produce and they could reduce inflammation based on the metabolites they will identify, like, for example, like nitric oxide, or even they will identify the pH. A normal vaginal pH would be 4.5 to 5, where if it is going to be little less based on any bacterial infection, this will kick in and they will help, the synthetic microbiome will help. Likewise, this is what I wanted to just tell you, but there is much of a thing. We can also repurpose many metabolites. For example, this is a metabolite acyl homoserine lactone, which is used for quorum sensing. We can use this quorum sensing as a very important characteristics to switch on or switch off certain mechanisms, in which we can take one advantageous gene sequence and metabolite to take advantage, or we can switch off them by which the metabolites do not go into it. Am I audible? Hello? 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 Okay. So the molecular therapeutics delivered by this engineered probiotic species, for example, a couple of molecular probiotic species that have come into existence from different wired species like E. coli strain Nisil or E. coli ECN, are mainly focused on many anti-inflammatory metabolites as well as certain bacteria that they can kill in bacteria. So coming to the synthetic life, so for what we have seen is programming the life. But whereas, is it really possible to to make a life synthetic? See, this is where the database comes into existence. We have found totally 60 million genes in the database, which are very unique, that have been taken from different bacteria, different living beings. Okay, so these 60 million genes produce 60 million unique proteins, which will have 60 million unique functions. So it is like now you pick and choose the 60 million genes to identify, and you get a flavor due to permutation combination in creating a synthetic genome. This is what Craig went ahead and did the first synthetic mycoplasma genitalium. And the one problem is creating the synthetic genome. Okay. So how do we make such a big genome? Do we have a cassette or a vector kind of a thing to do this? That is one big problem that is being circumvented. The other one is, okay, you do a very big synthetic microbiome. How are we going to make this microbiome genome boot up, make them alive, make them functional? That is another important thing, for which we need functional booting mechanisms that are there in different systems, which are being identified by these unique genes, and they are helped in the mechanisms, which currently there are a lot of concepts that are going on. It means to say that you create these 16 million genes in combination. It is like you create a software by which this software produces a hardware, which is nothing but a living system, for which we just resort to certain important computer programming like synthetic organism designer, where you could easily key in your sequences by which you could find and you can identify any synthetic organism with your own characteristics that you would. The reason how it is being done is being published for the first time by Craig Venter, where it's called a genomic transplantation in bacteria, where changing one species to another. As I was just telling you, the brewer's yeast, Saccharomyces cerevisiae, was able to produce human proteins. The reason being that we get the genetic code that converted into computer binary code. And this computer binary code is being fed into the system, a software which helps you to design the bacterium. So this is what is very much done in existing or making a new synthetic life into existence. But at the same time, we need to know that people are very scared about the synthetic biology. People thought initially when CRISPR-Cas came into existence that this is the biggest weapon in the world that is going to kill everyone. Now people think that synthetic life is more important. A person can produce synthetic bacteria, synthetic viruses like coronavirus or whatever it may be, that will kill or just devastate the whole population and the whole earth. So there are a lot of reservations for this, and there are a lot of ethical issues that are being currently going on in taming them for the best. So what I'm just showing you is the Craig Venter's Institute. I was talking about J. Craig Venter, who's a very pioneer, and he's called the father of synthetic biology. And I'm very fortunate to do one of my postdoctoral trainings there in J. Craig Venter Institute. What you see here is this is in Washington D.C., USA, and you could see here a little big fat. Now I'm not that much fat here. And this is my small group there at Loma Linda, where this is with students and faculties. A couple of them are involved with this kind of synthetic modification of microorganisms as well. So with this, let me just end my talk with a very big thank you that synthetic biology is the talk of the town now. But what we do really, making them synthetic plays a very vital role, and how we do them is very fascinating. And I should certainly say that India is now focusing on synthetic biology. We are bringing in synthetic microbes, which will be very useful for environment as well as for the human being, even in healthcare divisions, etc. Thank you so much.
Very much, sir, for your wonderful lecture about synthetic microbiome redefining host pathogen modulations. So you gave a brief introduction about gene editing, genome writing, genome reading in terms of genetic engineering, how the human microbiome is useful for the human being in terms of symbiosis and dibiosis. Really, it is a provoking thought, provoking research towards the scientific community and the students. Thank you. Once again, I am very much thankful to you, sir, on behalf of the organizing committee and Madurai Kamaraj University. Now I request the audience, if you have any doubts and clarifications, you may ask to Dr. Wilson Aroni.
So there are certain questions that, sir, how are metabolic flux I don't know. I'm just a couple of questions that I can see on the chat. I'm going to answer.
Yes, sir. Yes, sir. Please go ahead.
Okay. So many people are now bothered about the attendance. That is what I'm able to see here. I'm just focusing on number one. The biggest question is by Muthu Prasad. Could you please share the presentation? I should certainly say that thank you very much for asking this, but a couple of things are not being published so far, and people are doing their research on. Excuse me for not sharing it. What is the minimum gene number? It's by Dr. S.P. Nisha. What is the minimum gene number needed to synthesize a synthetic bacteria or a unicellular organism? See, this is, I should say that Craig Venter Institute had come out with this Mycoplasma genitalicum with the 564, if I'm correct, genes, which is the smallest and the second genome to be sequenced after E. coli. So that is why this Mycoplasma genitalicum is being focused on. I should certainly say that I'm not into creating synthetic life, but I'm focusing only on synthetic reprogramming things. But in my knowledge, somewhere around uniquely, I would suggest 800 to 1000 genes, basic genes to be there. And Angela, she had asked, how is microbiome metabolites related with the autoimmune disease? Yes, it is related with autoimmune diseases. For example, a lot of metabolites that have identified in are related to allergic reactions. For example, the end product of certain food that we take. For example, a person who takes alcohol, the end product of alcohol is nothing but formaldehyde. Okay, if you smell formaldehyde, it is a very big carcinogen. Imagine you drink alcohol and after that the next day your stomach is with formaldehyde. It means that you drank formaldehyde. So what is the end product of this formaldehyde are certain immediate broken down metabolites which also cause certain immune reactions or with low grade immune reactions. That is why people suggest not to take this kind of thing. This is one example which I wanted to tell you. The other couple of examples are there are a lot of metabolites which are there which causes autoimmune diseases like rheumatoid arthritis. We are focusing on this kind of rheumatoid arthritis thing, and we also found that certain the microbiome, detrimental microbiome produces certain metabolites. They cause predispose the human being to rheumatoid arthritis, etc. The next one is Shivani Sharda. How are metabolic flux estimation done? Only experimental or both computational also? Metabolic flux is done experimental as well as along with computational basis. We need to have the data, live real data, to feed into the computer to get the sequence of simulation that could happen. So we can do a time course experiment in which we can do a metabolic flux thing. A couple of things. Okay, Dr. V. Sugumar. Is synthetic biology the key to health? Yeah, every time people say something, when something new comes, it is very key to health. But ultimately, I should certainly say that the basic biology or the nature is the key to health, and then comes the synthetic thing that becomes key to health if the nature is not going to help you. So the other thing is Vimal Kishore Singh. Can you describe the synthetic microbes in obesity? See, I would give you one good example that people have identified a very important microbiome in the Japanese microbiome. And this Japanese microbiome, this bacteria exists in people who are very lean. And now people are patenting that microbiome, modified synthetic microbiome, and they are using it as a probiotic where, when you are going to take them as tablets, you will not become obese. This is one good example which I want to tell you. And the next is Rangesh Kannan. Is there any relationship between gut microbiota and human behavior? Yeah, it is there, which we call it as gut-brain axis. As I was just telling you, that when you compare a human being in India who is taking bhaji vada compared to a burger in US, entirely the whole system gets changed. Whole bacteria gets changed here. Certain bacteria are even related to mood changes. That is what I was just telling you. Where the bacteria that are being produced in your gut when you eat more of cheese or cheesy substance like what is a pizza, burger, etc. The next thing is M. Manikandan. If a patient is suffering from severe gastritis with ulcers, whether he can be treated with synthetic pathogens. Yeah, this could be done, but why do we want to go for a synthetic pathogen? First of all, number one, if a person is suffering from severe gastric ulcers, which is very common in our Indian setup, okay, people always burp whenever we eat anything, or even we don't eat anything, we burp, and these kind of things are happening because of your GERD kind of a thing, where a lot of probiotic formulations that are there rather than synthetic formulations. But I should also say that synthetic probiotics also came into existence now. Okay. So they are very good than the existing probiotic. But one thing you need to understand, if you are going to take a conventional probiotic, the function is very little. But whereas if you are going to make a synthetic probiotic, eat a synthetic probiotic, you are totally rewiring your system to make the system itself synthetic and beneficial. So it is very possible here. Debesh Chandra Bhattacharya, how can you determine the effectivity of a probiotic from synthetic microbe? Is there any metabolic flux analysis involved? I think this is a very big topic where no, I'm sorry, I will not be very crisp in answering it right now. We can determine the effectiveness of probiotic by synthetic microbes, because metabolic flux analysis is not going to be very different from using a natural bacteria versus a synthetic bacteria, which is going to be the same. Nadesan Balasubramaniam, how do you modify pathogenic virulent mechanism to a virulent by gene editing or writing its application in infection control? Yeah, it is possible. See, one of the things that we are currently doing is actually modifying the virulence by taking one gene out. Okay, which I told you one good example, or to make a chimera. Imagine if you are not going to focus on one gene, if you are going to take 10 or 20 different genes, you are making such a lot of variation in modifying the pathogenicity. So in that case, you make the bacteria immunogenic, but whereas not pathogenic. So this could be possible, and there are a lot of methods by which we modify them, which I have given a couple of them too. Vijay Pradap Singh, can you tell about the role of plant microbiome? I'm sorry, I can't comment on plant microbiome because I don't work on plant microbiome. My apologies. Abhijit, what are the risks of synthetically modified organism being released into the environment? Yeah, if you are going to modify the organism to suit your need, it means that the organism becomes effective. The gating mechanism will work only in the system where it is going to be used. Okay. If the system, the bacteria, you are going to throw it away, and if there is not going to be of a much variation, you needn't bother about that kind of modification at all. And at the end of the day, we talk about personal medicine. Synthetic biology and personal medicine is one of the unique things. So you don't literally goof around with these organisms into the environment. That is more important. So at this period in time, and I should certainly say that kind of an ethical issue in using synthetic modified organisms has not affected the ethical issue at all. Because as of now, there are genetically modified GMOs are there, GM plants are there. Everything has come. Whatever we eat now, maize and other things are GMOs. So I don't think it's a very big issue currently. Yeah, I think I'm pretty much covered all the these things. If there are no questions, I think.
Thank you, sir. Thank you very much for your wonderful clarifications towards the audience.
Thank you.
So really, once again, I very much thankful to you, sir, for your wonderful talk about synthetic microbiome redefining post pathogen modulation. So really, it is very useful to the students community and scientific community, sir. Thank you, sir.
Thank you very much. Thank you.
So audience, so the next talk going to start from 2015. So 2:15 p.m. Hello. Hello.
Yes, sir.
Audience. So next program will be start 2:15 p.m.
Yes, sir.
Yes, sir. Yes, sir.
Okay.
Okay, sir.
Okay, sir.
Okay. Session at 2:15 p.m. அவ்வளவுதான்.